All right, friends, welcome back. Welcome back. We're going to get going today with where we stopped last time, which is making acetals with carbohydrates. So if you recall, this is D-glucopyranose, and the squiggly line on C1 indicates that it's a mixture of alpha and beta anomers. And we're asking what happens when we put this molecule in the presence of an excess of an alcohol with catalytic acid, toluene sulfonic acid. We're removing water. As we discussed last time, first step, you protonate the C1OH group to turn it into a good leaving group. Then the neutral oxygen can kick off that good leaving group to get this intermediate, which we've called an oxocarbenium ion or an oxonium ion. And just to avoid confusion, recall that I said last time that your text might show this mechanism in a slightly different but fully equivalent way, which I'm going to draw for you here. Instead of having the electrons on the oxygen kick down and the leaving group leaves, your text will sometimes show the leaving group just leaving to get an intermediate that is in fact a resonance structure of the oxocarbenium ion that we've drawn above. So you could potentially be confused thinking these are two different mechanisms. They're actually exactly the same mechanism because they lead to intermediates that are in fact resonance structures of each other. So you should be comfortable with both of those representations. Then once we get to the oxocarbenium ion, the methanol nucleophile can attack the intermediate either from above or below. Now, normally, when attacking a carbocation of this nature or a carbonyl of this nature, both of those resonance structures, you would imagine that attack from above or below would happen with the same frequency for a planar intermediate. For glucose, the presence of the stereocenters at C2, 3, 4, and 5 mean that the top face of the oxocarbenium ion is not the same as the bottom face. So there can be a preference for one versus the other. Nevertheless, we would expect mostly to see a mixture of the two anomers. So what we'll draw next is just the squiggly line to the methanol, and then a proton on the methanol with a positive charge on the oxygen. And then in the final step of the reaction, we regenerate our acid by having the conjugate base of the acid deprotonate the methanol oxygen, and that's going to give us the methyl, what we will call the methyl glycoside of glucose. Glycoside is a term we use for acetals that are formed from the hemiacetal carbons of carbohydrates. So this is the methyl glycoside of glucose. Okay. Questions about that? That's pretty easy. That's a chapter 14 mechanism, something you should be familiar with, something you should have already mastered, but just in the context of a carbohydrate. Now let's talk about making glycosides where instead of having a small, simple, before we do that, I got to backtrack. On Friday, we should have talked about some additional carbohydrates besides glucose. So we're going to do that now. Glucose is the easiest carbohydrate to memorize because if you memorize it in its beta form, you draw the chair and then all the substituents at C1, 2, 3, 4, and 5 are all equatorial. And I told you last time this is beta D-glucopyranose. There are 2 or 3 common carbohydrates that are epimers of glucose. Epimer, recall, is a term that means a diastereomer that differs only in the configuration of a single stereocenter. So, for example, if we take the C2 hydroxyl and instead have it be up as opposed to down, the C2 hydroxyl is up, we call that beta D mannopyranose. And the open chain form would be called D-mannose. And mannose is found in a lot of carbohydrates in proteins that are excreted outside the cell, attached to proteins that are excreted outside the cell. And you can see we would call it the C2 epimer of glucose because it differs from glucose only at the C2 carbon, okay? So you need to memorize that one. That's mannose. You should recognize it if I show you the structure. You should recognize it if it's a subunit of a larger carbohydrate as well. Another epimer of glucose is the C3 epimer of glucose. This is less common than mannose or glucose or the other one that I will talk about. But if we draw everything the same except C3, then we would call this beta D allopyranose, and its open chain form would be allose, D-allose, and it is the C3 epimer of glucose. You won't really encounter allose a lot in biochemistry. It's not found a lot in biology, but because it's a simple epimer of glucose and because it's in between the two common ones, I decided to list it here. So mannose is the C2 epimer, allose is the C3 epimer, and then the last one we're going to memorize is the C4 epimer. So here the C4 hydroxyl is going to be up instead of down. So we would call this beta D galactopyranose, and its open chain form would be called D galactose. And as I said, yeah, it's the C4 epimer of glucose. So you need to have these structures memorized. You need to know the names as well. I've drawn the beta anomers. You should recognize the alpha anomers. And yeah, so the name galacto there will sound familiar, sound like galaxy. Comes from the Greek word for milk. Galaxy, we use that word for galaxy because of the Milky Way, right, in the sky. It's called galactose because this is a major component of lactose, which is a disaccharide. Galactose is one of the two carbohydrates found in lactose. All right. It turns out that nature can make galactose, biology can make galactose from glucose through what's called an epimerization reaction. We won't learn it here. You may learn about it in biochemistry. But basically, nature uses NAD+ to oxidize the C4 hydroxyl to a ketone. And then nature uses NADH to deliver a hydride to that ketone from the bottom face of the sugar, pushing the resulting OH group up. So a series of reduction and then oxidation again allows nature to convert glucose into galactose and vice versa. Yeah?
The gluco, mano, and galacto, are those roots, or I'm not sure what
Sure.
are they specific to kind of changes at the C2, C3, and C4?
That's right. Mano is the C2 epimer of glucose. Mano, that root refers to stereochemistry at C2, C3, and C4. And it's only at C2 for mano that it's different from gluco.
So it's at C2, C3, and C4 for mano?
No, don't misunderstand me. The mano term, or rather, I'm sorry, I'm not being clear. The mano term refers to stereochemistry at all three carbons. So you need all three of these to be just the way shown here for it to be mannose. But notice that mannose only differs from glucose at C2.
Gotcha. And then for, is it the C3 epimer?
Yeah,
Exact configuration from allo.
that's right. Yeah, must have this exact configuration at both 2, 3, and 4 to be allose, but it's only different from glucose at 3. Same thing with galactose, only different from glucose at 4. That's why it's most convenient to memorize glucose and then just remember that mannose is the C2 epimer, allose is the C3 epimer, and galactose is the C4 epimer. Yes?
And any of these on C1 can be alpha or beta up or down?
Yep, any of them at C1 can be alpha or beta. I've drawn them all beta here up, but it could be alpha down. Good. Okay, now, back to what I was saying earlier. Imagine you wanted to make a glycoside, but instead of using a small alcohol, you wanted to use the OH from another carbohydrate. So let me show you one of these that we isolate from nature. And we'll talk about it. We'll talk a little bit about its synthesis, mostly focusing on biology. So here is a disaccharide. Oops. That means a carbohydrate composed of 2 different individual carbohydrate subunits. And we'll say that this end of the sugar is still a squiggly line, meaning a mixture of alpha and beta anomers. We'll call this subunit A and this subunit B. This disaccharide is called cellobiose. It is a subunit of cellulose, which is made in plant cell walls, which is a component of plant cell walls. So first, take a look at subunit A. What would be the name of that subunit? It comes from glucose, right? Okay. How about this subunit? Also glucose. Good. Now, let's look at C1 of subunit A. What kind of a functional group is that? So we'll talk about stereochemical configuration alpha or beta in a minute, but what is the functional group? You can tell it's acetal because it's got 2 OR groups attached to it. Good. How about C1 of the second subunit B? What kind of a functional group is that? Hemiacetal, good. You can tell that it's hemiacetal because it's got an OR group and an OH group attached to the same carbon. Good. Sugars that have a hemiacetal are called reducing sugars. You'll hear that term sometimes. It's kind of an old-time term. Reducing sugar means a sugar with a hemiacetal. The reason they call it a reducing sugar is because hemiacetals are in equilibrium with the open chain aldehyde form. And that aldehyde can be oxidized in an experiment, and it can reduce, as it's being oxidized, it can reduce something else. In fact, there's an experiment, I should have ordered the demo today. There's an old-time qualitative experiment called the Tollens test, where you take a silver 1 salt and expose it to a sugar, and if the sugar has a hemiacetal in it, the aldehyde form of that sugar will actually reduce the silver from silver 1 to silver metal. And if you do it right, it will coat the inside of the flask with a silvery mirror-like structure. It's kind of fun. We do it at our chemistry magic shows. But it used to be a way to test, do you have a hemiacetal in your molecule? And that language has still persisted in carbohydrate chemistry. So for example, people will talk about sugars having directionality. This is the reducing end of the sugar, that's the hemiacetal end. This is the non-reducing end of the sugar. This is the end that lacks a hemiacetal. This portion is not, this is an acetal. Acetals are stable to various conditions. They are not in equilibrium with open chain forms, so an acetal is not a reducing sugar. Okay, let's talk about the linkage between C1 of subunit A and C4 of subunit B. And we'll talk about how people talk about this linkage. I'm just going to highlight it. That portion there is called, we could call it the acetal linkage, but we're using the term glycoside for a carbohydrate-based acetal, and therefore we call this a glycosidic linkage. And we describe that glycosidic linkage with the numbers of the atoms, the numbers of the carbons that are involved, starting with the acetal end of the sugar. So in this case, that is C1. And then we draw an arrow pointing to C4. So we'll describe this glycosidic linkage as a 1,4 linkage. But we're not done because we need to describe the stereochemical configuration of C1. In this case, it's beta because the C1 oxygen is up on the same side of the ring as the C5 hydroxymethyl group. So we will call this a beta 1,4 glycosidic linkage. Okay, questions? Yeah?
Are there situations in which we get similar molecules but they're alpha?
You bet, and I'll draw you another one of those in a little bit, yes. So you can get the alpha 1,4 linkage as well. Now, looking at that, you might have some questions or concerns about how you might make this molecule. Because if you tried to do this via the chemistry that we showed above, one of the things you'd be worried about, presumably you would have an oxocarbenium ion like this. This is the oxocarbenium ion of glucose. This is just an intermediate taken from acetal formation above. And then you would need a nucleophile to attack that oxocarbenium ion. And you want that nucleophile to be this C4 hydroxyl. If you were doing this in the lab, what problems do you foresee? Go ahead.
You could have self-attacking self.
Okay, so if this is subunit A and this is subunit B, you're worried about perhaps this OH on subunit A attacking the A versus subunit B. Okay, good. What else? Go ahead.
You've got a lot of alcohol or OH groups on B.
Right, alternative OH groups. You got 4 other OH groups that could be nucleophiles there. Okay, good. What else? Go ahead.
You either add on the top or on the bottom.
Right, you got stereochemistry problems. We could go above or below, getting the beta anomer versus the alpha anomer. So 2 key problems in the lab are regioselectivity and stereoselectivity at the anomeric carbon when we want to make a glycoside. And your text doesn't talk about this because this is sort of graduate level stuff. It requires a ton of protecting groups, and sometimes you need a different kind of protecting group at C2 versus C3 versus C4. So you need a big library of potential protecting groups to use. And then controlling addition stereochemistry up to the oxocarbenium ion is challenging. And there are a lot of techniques developed for doing that. Some work better than others. In a lot of cases, you have to purify the alpha anomer away from the beta anomer and hope that you can separate them by chromatography. There are people who are very good at doing this. Professor Paul Savage in our department is one of the world experts on carbohydrate synthesis. His lab can basically make any carbohydrate that you want, and people around the world collaborate with him for really interesting projects. It's challenging and it's really, really tough work, and fortunately we're not going to talk about it at all. But if you wanted to make this disaccharide and none of the alternatives, you would worry about the potential mixture of molecules. And in fact, if you've ever made caramel or rock candy, this is sort of what you were doing. You put table sugar, and we'll talk about table sugar later today. You put table sugar, a little bit of water, and you start heating it up. No protecting groups, and OH groups start attacking oxocarbenium ions, and you get lots of linkages between many of the OH groups of a carbohydrate with other carbohydrates. It's highly branched. It starts to get more viscous. If you continue to cook it, it will start to get to the point where it hardens when it cools down, and then you've got rock candy. So that's uncontrolled glycoside formation. How does nature do it? If we do it in the lab, we need a lot of protected groups and purification. Nature doesn't need that. And so what I'll give you is just a quick sense for how nature does these, makes glycosidic linkages. It's going to be pretty general. We're not going to refer to any particular enzyme, but I'll just try to give you a brief sense for how it works. So the class of enzymes that does this are called glycosyl transferases. You'll learn about them in biochemistry. And nature solves the regioselectivity problem with the enzyme active site. So the enzyme active site is a pocket in an enzyme that brings two things close together. It brings subunit A close to subunit B in exactly the right orientation such that it's only the C1 hydroxyl that can attack subunit A, and it can only attack from above. So let me show you an example of how nature does this. The molecule that receives the nucleophile is actually not glucose itself. It's a version of glucose in which the C1 hydroxyl has been replaced by a good leaving group. And that good leaving group is called UDP, which stands for uridine diphosphate. I'm going to just abbreviate it real briefly here. So here is the oxygen that's attached to the C1 of glucose. The diphosphate portion is this phosphoanhydride here. There's usually a magnesium dication associated with that phosphate to help it as a leaving group, and then that oxygen is attached to the 5' hydroxyl of ribose, and then here is the base uridine. We'll talk about its identity a little bit later in our chapter on nucleic acids, but for our purposes now, you can think of this as a good leaving group. Okay? And and it turns out that the enzyme sort of prevents the UDP leaving group from leaving on its own to give an oxocarbenium ion. So the leaving group is there when the nucleophile attacks. One of the ways enzymes control stereochemistry and accelerate rate is they change the mechanism. So notice that the mechanism of glycoside formation in acid catalysis involves the leaving group leaving to give an oxocarbenium ion. In the enzyme, the leaving group doesn't leave until the nucleophile attacks. That's going to help control stereochemistry. And let me show you how. Here is subunit B, and the enzyme holds on to subunit B such that only the C4 hydroxyl is close to where it needs to attack. And the other thing the enzyme does is it uses a basic group. So this ENS B refers to a basic amino acid side chain in the glycosyl transferase, which removes the proton on the C4 hydroxyl of the thing we want to be the nucleophile. As that proton is being removed, the oxygen then attacks at C1 and the leaving group leaves. That mechanism is SN2. Do you see how that would control stereochemistry at C1? What happens in SN2? You get backside attack, and then what happens to the stereo center? It inverts, right? So this used to be alpha-linked UDP glucose, but the nucleophile attacks from the top face, and that's why you're going to get the beta linkage. And there are various glycosyl transferases that can control whether you get the alpha versus the beta linkage, depending on how the nucleophile is positioned. So the enzyme solves the stereoselectivity problem by making the mechanism SN2. It solves the regioselectivity problem by only deprotonating the right OH group, and it controls that by positioning the molecule in the right place such that only the C4 group is close to the base. Okay, so there's not a lot to memorize there. I just want to show you how nature is able to do this, because you might have been worrying about, well, how do you prevent, how does nature do any of this without generating rock candy? And the answer is enzymes are amazing. Okay, so what we're going to do now is talk about other types of disaccharides and more complicated carbohydrates. And your goal here is to identify the subunit identity, glucose, mannose, galactose, and then to identify the glycosidic linkage, beta 1,4, alpha 1,4, so on. Go ahead.
So you mentioned caramel as well, and I'm sorry, it's a little bit off topic, but is caramel just with like your table sugar and then lactose from milk, or?
I'm trying to remember how it works. I think when Amber's got a recipe for salted caramel cupcakes, which he hates making because I think you have both. I'm trying to remember how it works, but I think it's butter and sugar together at the beginning. No, I think it's sugar first until it starts to caramelize. Then you add butter and it starts to spit at you because the lipid melts. Really hot sugar gets hotter than boiling, and so when you put water or liquid in it, it tends to boil and spit all over the place. Then at the last minute you put in cream, and again you're going to get hot oil splatting at you and cream. So yes, caramel is more than just the sugar. You're putting in, I think, some fats and then some dairy as well. You have to go to food scientists to know more about it than I do. I'm just talking about the carbohydrate portion, so that's interesting. Yes?
Isn't there also glucose, like, since sugar is typically glucose plus glucose?
That's right. I will show you the structure of table sugar in a little bit, and you'll see that it's composed of a glucose subunit and a fructose subunit. Yep. Okay, so let me show you now something that's going to look a lot like cellobiose, but the linkage is going to be different. This is maltose. And maltose is a subunit of starch, a disaccharide subunit of starch. I'm going to continue to show the hemiacetal end as a squiggly line that can have both configurations. Okay, so subunit A, subunit B. A is what? What carbohydrate is that? Glucose, easy, right? What about B? Also glucose, okay. What is the identity of the glycosidic linkage? Okay, the configuration is definitely alpha. Then what are the correct numbers? You always start with the anomeric carbon 1, and then you end with the alcohol carbon, which is what? 4. Yep, alpha 1,4. So maltose has the alpha 1,4 linkage, which your body does have enzymes to digest. Cellobiose has the beta 1,4 linkage between glucose, which your body doesn't have the enzyme to digest. Okay. So let's see. Now I guess I should show you the structure of starch, which is a much, much longer carbohydrate, but you'll see pretty quickly how it's based on this basic maltose 2-unit alpha 1,4 type of linkage. So here is one sort of maltose subunit of a carbohydrate. And I'm going to do the unthinkable here and use copy and paste so that we don't have to spend all day drawing glucose. You don't have to copy this down, it'll be in your notes. So if I take this and, sure, maybe I'll just put it there. Copy, paste, copy, paste. And then do squiggly lines. This molecule is called amylose. The squiggly lines mean that the molecule can extend basically indefinitely with additional glucose subunits. And notice that they are all alpha 1,4. Okay. Oh, that's amylose. There should be an S in there. Okay. And your body has an enzyme called alpha glucosidase. It's called glucosidase because it's good at hydrolyzing the alpha glycosidic linkage between glucose subunits. And this starts to work even as you start to put food in your mouth. So if you want to do a semi-gross experiment, you can go to Walmart and buy some saltine crackers for $1 or whatever, or $5 with inflation or whatever it is. But and then put one in your mouth, start to chew it, let your saliva, but don't swallow, and then just suck on that soggy cracker for a while. It's disgusting, but over time you'll notice a little bit of sweetness start to grow in. And it's not going to be as sweet as table sugar. It turns out glucose is not that sweet. There's a ranking of carbohydrates, a sweetness index, and glucose isn't that high on it. Sucrose, table sugar is. So it's not going to taste like candy or table sugar, but you will start to detect some sweetness, and that's individual glucose subunits being hydrolyzed off the sugar. Yeah?
Is like a shorter version of starch?
Yes, this is a component of starch called amylose. There are maybe a slightly more complicated or branched starch molecules, one of which I'll draw here. So I'm going to copy this amylose chain. It turns out that in some starches, the C6 hydroxyl gets modified with a glucose molecule, and then additional alpha 1,4 linkages branch off of there. So the carbohydrate I'm about to draw is called amylopectin, and it has a linkage between the C6 hydroxyl of glucose and C1 of another amylose chain. So I'm going to try to show this kind of strange. Let's see. Nope. Twist around, spin, and I'm going to need to erase that one. There we go. So that's sort of a branch point, and at the branch point, this is a linkage between, it's an alpha linkage between C1 and C6. So we call that an alpha 1,6 linkage. Sometimes people use parentheses, sometimes they don't. It doesn't really matter. Alpha 1,6 linkage, and that's a branch point. And you can imagine that branching would change the properties of the starches. Anyway, okay, other questions? Okay, let's do a few more. Let's do lactose. So this is a component of, this is a carbohydrate found in dairy. Okay, it's like maltose and cellobiose, it's a disaccharide. So subunit A and subunit B, what is the identity of subunit A? That's galactose. You can tell it's galactose because the C4 hydroxyl is up. Galactose is the C4 epimer of glucose. People will abbreviate galactose with the 3-letter code GAL. You're not going to need to memorize that, but you may see me do that so I don't have to write out galactose. Then what about subunit B? What's that? That is glucose. Easy. All the groups are equatorial. Okay, what about the linkage between the two? What would you call that glycosidic linkage? Beta, because at C1 the group is up, so that's a beta configuration, and it links 1 and 4. And so sometimes people will write out the actual structure of lactose in this way. It's a galactose linked to a glucose via a beta 1,4 linkage. Okay, you guys ever heard of lactose intolerance? Okay, what is lactose intolerance? Go ahead. Okay, so yeah, you can't digest the lactose, meaning you can't hydrolyze the beta 1,4 linkage between galactose and glucose, but some bacteria in your gut microbiome can. And they do that, and it generates unpleasant GI symptoms, right? Which some people are like, whatever, ice cream is good, I'm just going to power through, right? And it depends. There's some foods that have more lactose than others, and you sort of have to play around with it. Okay, let's talk a little bit more about lactose intolerance and why that might happen. So, when you were an infant, your diet was exclusively human milk. And human milk has an oligosaccharide in it that looks a lot like galactose. I drew it for the 9 o'clock class, so I guess I'll draw it for you. You don't need to memorize it, but this is one of the oligosaccharides that's present in human milk. It shares this saccharide. I can't remember how to spell saccharide, so I think there's an H in there somewhere. It shares this basic lactose and glucose structure with the beta 1,4 linkage, but in human milk, the C2OH is modified with an additional carbohydrate. This is a weird one in the sense that it doesn't have as many OH groups as glucose does, and the C5 group is down instead of up. So this is actually L-fucose. It's one of the few L sugars that nature uses. But in any case, human milk oligosaccharide contains this same beta 1,4 linkage. When you were an infant, you needed to be able to digest that, and so you have an enzyme that's called beta galactosidase that hydrolyzes the beta linkage, the beta glycosidic linkage between galactose and glucose. Okay, and so you were happy as an infant, you got nutrition. So long ago, before humans domesticated animals, and long before a large number of us regularly got nutrition from dairy, after you stopped nursing, you didn't necessarily need to hydrolyze galactosidase anymore. And so in most humans, the expression of that enzyme in your GI tract would fall off as you came closer to adulthood. Because again, the human diet back then didn't involve a lot of milk. Well, as humans migrated and moved, I read this book called A Brief History of Everyone Who's Ever Lived. It was really fascinating. As humans migrated out of Africa into other parts of the world, and as animals started being domesticated in Europe, it was really cold. And you couldn't grow things a lot during the winter. But humans learned how to make and store dairy products. So cheese, you can store for years, right? And cut off a hunk of cheese and eat it when you're cold in the winter. Butter, you can also store. And so there became an advantage for humans to get nutrition from dairy. And about that same time, there was a mutation that then expanded because it provided a selective advantage where some human adults were able to express beta-galactosidase and hydrolyze lactose. Again, a single mutation that expanded into a population because of a selective advantage. So people in Europe, because they got nutrition from dairy, stopped being lactose intolerant, whereas most other humans continue to be lactose intolerant. So to this day, if you have predominantly European ancestry, you are likely not lactose intolerant. If you don't, you may be lactose intolerant. Sometimes it doesn't work out that way. My son Jack, the missionary, is lactose intolerant, but he mostly powers through because chocolate milk is good and just deals with it. But if you don't want to deal with it, there are products out there, Lactaid, which actually you take some of the enzyme, you swallow some of the enzyme that can hydrolyze the lactose, and you do that on sort of a dose dependent when you're going to drink milk or eat ice cream kind of thing. Anyway, interesting story, but it all has to do with stereochemistry and being able to hydrolyze specific linkages. I ought to show you the structure of table sugar. So as was alluded to earlier, sucrose is a disaccharide of two subunits, glucose, which you've seen before, and fructose, which you haven't. Fructose is a regioisomer of glucose, not a stereoisomer. And in our chapter on metabolism, we will talk about how to get fructose from glucose. But here is the glucose subunit. And then here is the the glucose subunit, and here is the fructose subunit. And fructose is a 5-membered ring containing sugar. We're drawing it in sort of a pseudo chair. There's a down OH group here, and then the C5 hydroxy. Oops, I did it wrong. Sorry, this should be, that's carbon 1, 2, 3, 4, 5, and 6. And this is the glycosidic linkage between glucose and fructose. So take a look at that structure. Tell me what you notice about it that is different from the other carbohydrates, the other disaccharides that I've showed you today. Okay, there is a 5-membered ring, so that's one interesting difference. What else? What's that? Okay, there are some axial groups on that ring, though probably the 5-membered ring, it doesn't necessarily adopt one chair. It's quite flexible. So what else do you notice that's different about this one? And let me just show you lactose as an example. Go ahead.
So 1-2 linkage?
Yeah, it's a 1-2 linkage instead of a 1-4 linkage. Good. Okay, one more thing. What does galactose have in terms of functional groups that sucrose, lactose have in terms of functional groups that sucrose doesn't have? Good. The hint is hemiacetal. Notice lactose has on C1 an OR of the glucose subunit, an OR group and an OH group. That's a hemiacetal. So lactose is what we would call a reducing sugar because one end could be oxidized by silver. In sucrose, this carbon is an acetal. There are two OR groups on that carbon. And this carbon in sucrose is also an acetal. There are two OR groups. There are two acetals. In this sugar, the two anomeric carbons are linked to each other. Therefore, sucrose is a non-reducing sugar. If you tried to do the Tollens test with sucrose, it wouldn't reduce the silver 1 salt to silver metal. I'm not necessarily sure how to talk about this glycosidic linkage, because in glycosidic linkages, we always start with the acetal carbon, but in this case, there's two. So the configuration of C1 of the glucose subunit is alpha. What do you think the configuration of the subunit, of anomeric carbon 2 of the fructose subunit is? To do that, you look and see, is it on the same side or opposite side as the hydroxymethyl group on C5? It's the same side, so we would say it's beta on that side. So I'm not sure how you write this glycosidic linkage. Is it alpha 1 to beta 2, but why should the arrow point just one way? Because they're both anomeric carbons. Maybe it should be like that. I don't know. And you're not going to need to describe that for sucrose. I'm not going to ask you a question about that. I'm not going to have you memorize the structure of sucrose. But you should be able to look at the structure of sucrose, identify the glucose subunit. You don't need to recognize the fructose subunit yet, but you should be able to tell that there's no hemiacetal, and therefore it's a non-reducing sugar. Okay, there's one more carbohydrate I want to show you. This is a variation of glucose, and it's called glucosamine. Usually we drop the E after the S, glucosamine. You'll have heard of this before. Some people take it as a supplement to improve joint health, and the idea is that it's a component of the extracellular matrix, but I'm not sure what the data are on it, so I don't know what to tell you there. It's like glucose, but the C2 hydroxyl group is replaced by an amine. It's only ever the C2 hydroxyl group. So if you hear glucosamine, you don't need to worry about where the amine group is. It's always on C2. In a lot of carbohydrates, that C2 amino group gets modified as an amide with an acetyl group. So what I've drawn for you here is called N-acetyl glucosamine. The N in front of the acetyl tells you where the acetyl group is, and that's why it's there. An abbreviation for this that people will use is sometimes called glycinac, which is shorter than saying N-acetyl glucosamine. This is found as a subunit in chitin, which is insect shells. And I'll show you a 2 carbohydrate subunit of chitin. It's called chitobiose, and it involves 2 glycinac sugars linked together in a beta 1,4 linkage. So 2 N-acetyl glucosamines linked together . And this is what insect shells are made of. And it's interesting that the addition of that acetyl group rigidifies the carbohydrate so dramatically relative to just if there were OH groups there. Okay, so that, let's see, that concludes our chapter 24 on carbohydrates. We will start lipids next time. See you Wednesday.